So now that we better understand regulatory elements and know about kind of where they occur, what their impact is, some of the trends that we can see in their evolution and their change, we're going to give you some specific examples to reinforce some of those points. The first point that we want to reinforce is this conservation of functional elements among widely divergent taxa. And what I mean by a functional element is anything that's really important for a critical developmental process. So this could be a regulatory element, but more commonly it's a gene and a timing and level of expression for that gene. So genes show remarkable conservation, and the way that those genes, those really important genes, are used often show remarkable conservation also. Now, to understand this, we need to understand a fairly simple concept, and then we can use this as our null hypothesis. And this concept is that as DNA diverges, we would expect it to start to become more and more different. So random mutations build up in DNA sequences and base pairs, and those sequences diverge. So if we were to line them up and compare them initially, they would be 100% identical. But as new mutations occur, those DNA sequences become more and more different. However, there is a plateau effect, or a level at which those DNA sequences stop becoming completely different. And to understand this, it's a rather simple game of probabilities. There are only 4 possible nucleotides in a DNA sequence: adenine, thymine, cytosine, and guanine. And so as mutations occur and as DNA sequences become more and more different, there becomes a point where we can essentially treat them as if they were randomized data, because they become so different, so many mutations have occurred, that even though they have originated from the same homologous piece of DNA, there no longer is any connection or very little connection correlation back to that original homologous piece of DNA. They've essentially become randomized sequences of DNA. And so the question is, if we randomly generated 2 DNA sequences and then just simply line them up, how similar would we expect them to be? And by doing that, we can actually answer this same question. After divergence, how similar would 2 homologous regions of DNA? And so it's a rather simple prediction. For any one site, the first site, for instance, there's a 1 in 4 chance that if we randomly generate a DNA sequence, we're going to have the same thing, since there are only 4 possibilities. So we would have a 25% chance at any one site. And so, on average, 1 out of every 4 base pairs in randomly generated sequences of DNA are going to match one another. Okay? And so we can do the same thing. After homologous sequences of DNA diverge, then we would expect there to be only about 25% similarity across them. But it's still, even 25% similarity is a rather high level of similarity. Now, this assumes complete neutrality, meaning that there's not some other force acting on those random mutations. Some of them are kept, some of them are lost, but it's random. And there's not any natural selection force that is either working to conserve DNA sequence, that would be called purifying selection, or there's not any sequence that's working to diverge them. And even divergent ones still are going to be roughly 25% because we're still working off of the same basic 4 base pair language, if you will, this genetic language. Okay, so keep that in mind. Now, anytime we see a level of similarity that is higher than 25%, significantly higher, then we would expect there to be some other explanation. Then there must be some sort of natural selection force that is working on that piece of DNA. So let's look at some examples, and let's look at conservation among excuse me, the regulatory portions of genes. And we're going to look at conservation at the level of the DNA, but also at the level of larger functional units, right? These functional elements, including regulatory elements. And we're going to focus on regulatory elements because that's what we're talking about in this unit. Now, if there is complete neutrality, then we can use that level of divergence to estimate time since those sequences shared a common ancestral sequence. And this is called the molecular clock. We talked about this in class. But realize that the molecular clock begins to run into a problem as we reach saturation. Now, what I mean by saturation is if we're having 2 mutations at the same location, maybe in the same sequence or maybe at the same location in different divergent sequences, we're going to be missing some of the events that have occurred. So for instance, if I have the sequence ATCG, and then that diverges into two lineages, initially they have the same sequence, ATCG. But then let's imagine in one of those lineages that G mutates into a C. So if I line up those sequences, I say, oh look, there's one difference, I estimate the mutation rate, and I know about how long on average it takes for mutations to occur. I can estimate divergence time. And so we begin to accumulate mutations in that DNA sequence, and I use every mutation as a measure of how long it has been since those sequences diverged. Pretty straightforward, right? But now imagine eventually, just by chance, there's a mutation at the 4th position in the other sequence of DNA. So it's ATCG. The other one had mutated to ATCC, but then I have the 4th position that mutates to ATCA. When I line them up, I'm still only counting one difference between those two sequences. I've got ATCA and ATCC. And I say, oh, look, there's one mutation. They must have been diverged X amount of time. But in reality, that one difference that I can only see the one difference, but in reality it represents two separate mutations. Now that's what I mean by saturation. And the plateau for saturation is at 25% similarity, or if you'd like the other way around, you can look at it as 75% difference between mutations, or between sequences. Now as we reach that plateau, eventually we get to the point where there's been so many changes that there's no longer any estimate that's valuable to estimate divergence time, because they're saturated and we could have had them reach that saturation point recently, or it could have been at that saturation point for many, many thousands of generations. So at saturation, there's no more useful data anymore. But as we approach saturation, we can actually correct a little bit and use models of evolution to help us correct for the expected levels of saturation as we're approaching the plateau point, but not quite there. And that gets harder and harder to do the closer you get to that plateau point. So the molecular clock is very useful if we are not yet close to the plateau point, and it can still be used, but with some caveats and some calculations as we begin to reach that plateau point. Now, if we are seeing that after many, many thousands of generations, maybe at the point where we should have been approaching the plateau point, but we have not yet reached that, then we know that there's some other force going on, that because in the long term, randomness is predictable over large sample sizes. Okay, so we can estimate divergence time for neutrally evolving sequences, but if there's some other force going on, then we can no longer use that neutral theory to estimate. And so we could line up sequences of DNA, and if we recognized, and these are homeotic genes from a single species, actually I think this is the same gene from many species, and we can recognize areas that are under very, very strong purifying selection. This is very constrained. No changes at all at the amino acid level across large chunks of this, and then only a few changes here and there across other parts of it. So that represents a fairly strong trend, and if this is over hundreds of millions of years, then we can say that, hey, there's some other force going on here. So this is looking at the gene, but what happens when we look at the regulatory elements that control genes? Do we see those really same strong levels of conservation? Okay, so here is a regulatory element for two species of Drosophila for the same gene, and we've highlighted areas that are identical. I notice that there's a lot of divergence, a lot of differences that have occurred. And so we have nearly reached saturation point for this, so we would expect them to be very differently regulated because they are so divergent. There are areas that are missing from one of the species. There are areas that are apparently still homologous, but no longer share matching sequences. So notice through here, they're lined up, but there's only a very few matches in there. Okay, so again, maybe 1 out of 4, that seems to be kind of at this saturation point, and we would expect there to be very little to no similarity in the regulatory elements there. Okay, so let's look at the dynamics and let's see what we see there. So the dynamics of sequences change in these cis-regulatory elements. Now to do this, we really need large sample sizes, and we're getting better. We're getting large data sets. We're being able to identify regulatory regions, but it's still tricky sometimes to identify regulatory regions just by looking at the sequence. We need to do some more functional studies, some more molecular biology type things to actually identify and target these regulatory elements. So although we are getting better data sets and we can make some extrapolations, there's still some work that needs to be done in this area. But Drosophila, as you might expect, is very well known and very well characterized for many genes. So here is a map of the upstream regulatory area for even-skipped, which is one of those developmental genes. It's a segment polarity gene, I believe. Maybe it'd be a little bit before that in the sequence that we learned for the Drosophila developmental genes. But it's a very important, crucial gene in the early development of Drosophila. And so notice that we've got 4 different molecules that all play a role here, and they have different binding sites. Now the names are not really important. You might recognize some of them like bicoid. So notice there are 5 different places that bicoid can bind, and some of those are actually a competitive binding site with another gene. So there's this complex way to control this gene. So we've got 4 genes and a fairly standard, a fairly well documented and well described regulatory regime for this gene. Okay, and so our question is, if we were to look at this in other organisms, do we see the exact same thing? Okay. Now, when it turns out that when we do this, this gene is essentially expressed the exact same way in all 5 of these species. So we've got Drosophila melanogaster compared to 5 other species here. And we're looking at whether it's completely preserved perfectly, we call that a P, whether it is strongly conserved, so pretty much the same thing but maybe 1 base pair change, whether it's weakly conserved, right, or whether it's just gone. And despite there being, these are closely related species, and despite there being exactly the same expression domain for this gene, there have been some changes. Now some appear to be more important than others, that changes have been removed. So these are really good, strong signs of purifying selection, right, where everything is identical. But in others, there's been a lot more flexibility. And so there's a little bit of a paradox here, where we've got complete conservation in the way that the genes are expressed, but yet we have some flexibility and variability in the regulation. And really the way that we look at this now is that there are often multiple ways to accomplish the same expression profile. And so often as random mutations occur, we're kind of exploring those, and if it's a really critical gene, it needs to be expressed the same time, the same way, and there are going to be very few mutations that change the expression domain of that gene and are still beneficial. So the expression can be conserved, but yet we might have a different regulatory regime, a different complement of these regulatory elements that get us to that same level of expression. And that's actually a very, very common thread that we see. So much so that there's even some extreme examples of this. And so we can think of some of these changes as compensatory mutations, where a change might occur that makes a slight difference, but not enough to really destroy the outcome. And so it works okay and it works all right, but then a second change occurs that makes up for that first one and kind of restabilizes it. And so that allows for some flexibility in evolution and some exploration of all of the different possibilities and still maintain the integrity and the expression levels of that gene. Okay? So here's an extreme example. Now, most of the time we'll see some variation and some preservation like we saw in the Drosophila example. But there are some examples of really extreme divergence in regulatory elements, and yet with some very similar outcomes. So on average, about 30 to 40% of the regulatory elements have changed, either a little bit or sometimes a lot. And so on average, between these species where we've got good, complete data sets, we see still a fairly significant amount of conservation, much more than we would expect just by chance. And so there's obviously some things going on, but still it's a fairly wide level of divergence. But again, some extreme examples. So we're going to look at some of these extreme examples where it's nearly completely 100% different, and yet we still have the same end result. So sometimes there are ways to do things very, very differently and still end up with the same conserved result. And this sea urchin is a very clear example of that. So here we have two different species of sea urchin with some slight differences in where things are expressed, and we won't get into all the details. But if you look at these two developmental sequences, you notice they're nearly similar. The red arrows on the right illustrate a little bit of a difference of expression of genes, but notice that the outcome is roughly the same. So for all intents and purposes, these have identical developmental sequences, and we're looking at this one particularly important gene. Okay? And when we look at it for species 1, all of these little squares and ovals here represent the regulatory elements that are controlling this developmental gene, this really important gene in species letter A. These great names, right? They Strongylocentrotus purpuratus. Sometimes I think that these species are named, the scientists were trying to troll future generations of researchers. But anyway, Strongylocentrotus purpuratus has this really well-defined and fairly complex control mechanisms for controlling this important developmental gene. And it results in a very stable and fairly standard sea urchin embryos as sea urchins go. Well, when they looked at this close relative, Lydiochinus variegatus, they noticed that although there was a little bit of conservation, so up front, these were much the same, that they could not find any of the same regulatory elements upstream of that. So again, not quite 100%, but notice like maybe 90% of the regulatory element region has changed, and yet we have the exact same role. Now, in this one, at the time when this was published, that doesn't mean that there aren't any regulatory elements there. It just means that they were unable to identify them as the same regulatory elements that were there for this other species. And so something else has maintained the same basic level and region of the expression for this gene, but the regulatory elements have changed. So it turns out that there's a high degree, again, on average about 30 to 40% turnover of regulatory elements while still maintaining the exact same domain and levels of expression. So that provides lots of flexibility in those changes. It's kind of the old saying, there's more than one way to skin a cat. Well, it turns out there's more than one way to develop a fruit fly. And some of it might be just variation, but there's certainly, for this early developmental stage of the sea urchin, there's a very, very different way to end up with basically the exact same outcome. So that's a very common trend that we see across these regulatory elements as we begin to define them and analyze them. And we'll look at some more examples in the next discussion.